Hello everyone, welcome to Patho Excellence. Today we are starting a new series 100 plus histopathology patterns. So, why these patterns are necessary? Because histological diagnosis depends on four major features: architectural pattern, cytology, stroma, and spatial stain and IHC. So, among these, architecture is usually the first clue. Now, if we see pattern, histopathology is all about pattern recognition. So before immunohistochemistry, before molecular studies, and sometimes even before reading the clinical history, an experienced pathologist first recognizes the architectural pattern under low power. Suppose a Zellballen pattern is seen, it immediately suggests a paraganglioma. Cribriform pattern, adenoid cystic carcinoma. Herringbone pattern points toward fibrosarcoma, while Indian file infiltration, a classic clue for lobular carcinoma of the breast. So this entire series is designed to help all the students and residents master more than 100 classical histological patterns. One pattern at a time with HE slides, simple diagram, memory tricks, etc. So whenever you see other slides, questions must be there before diagnosing slides like, are the cell arranged in nest? Are they forming glands? Are they spindle shaped or having surrounding vessels, or the tumor cells are they surrounding vessels? Are they forming rosettes? And once you recognize the pattern, the diagnosis becomes much easier. So in this series, we will discuss architectural patterns, all these different patterns, neural patterns like different rosettes, spindle cell patterns, vascular patterns, growth patterns, stromal patterns, spatial cell arrangement, necrosis patterns, spatial tumor patterns, and some classic patterns in specific tumors. So we are uh, discussing these all patterns so that you can remember the diagnosis on the basis of pattern, and once you recognize pattern, there is a clue to define the diagnosis. So in this first episode, epithelial patterns, we are going to discuss most common pattern of neuroendocrine tumor: Zellballen pattern, nesting pattern, trabecular pattern, organoid pattern, and solid sheets, right? So let's start with the most common pattern, Zellballen pattern. So by definition, Zellballen is the word, German word, means balls of cells. So it is an architectural pattern in which tumor cells are arranged in a well-defined rounded nest and separated by delicate fibrovascular septa. This is most common identification point, and along with this, these nests are surrounded by a specialized supporting cell called sustentacular cells. So in low power view, you will identify multiple rounded islands of cells. These are mostly uniform, having thin fibrovascular septa, rich capillary networks, and sharp border, right? So overall appearance resembles grape-like clusters. So here you will see well demarcated borders. Now nests are there, and the specialized cells are sustentacular cells. They are the supporting stromal cells. And if you see the cells are round to oval with prominent nucleoli and having granular red or purplish cytoplasm, or some of the cells may have vacuoles. So This is called Zellballen pattern, rounded nest, fibrovascular stroma, and surrounded by this elongated sustentacular cells. So, this is how you can identify in high power view. Now, why this pattern form? Because neuroendocrine cells naturally grow in nest, and each nest is surrounded by basement membrane having rich vascular network and sustentacular cells. So remember, sustentacular cells, vascular network and basement membrane. Now, what are the tumors showing Zellballen pattern? So classic is pheochromocytoma and second is paraganglioma. Occasionally extradural paraganglioma may show this type of pattern along with carotid body tumor. Now, differential diagnosis. We have to differentiate it from nested neuroendocrine tumor, alveolar soft part sarcoma and metastatic RCC. So, here helpful is IHC marker. So, tumor cells mostly chromogranin positive, simnitophysin positive, INSM1 positive, and these sustentacular cells are beautifully positive with S100 marker, right? As along with the SOX10 also positive. So, this was about Zellballen pattern. Now, second is nesting pattern. So, by definition, nesting pattern is a pattern where tumor cells are arranged in discrete nests or islands and separated by connective tissue. Unlike Zellballen, these nests are less regular and not necessarily surrounded by sustentacular cells. So, there is absence of sustentacular cells in this pattern. So, here you can see this is a pattern showing nest, multiple different size nests are there, and these are separated by connective tissue. So, this is again low power view and high power view shows numerous islands and clusters and nest with fibrous stroma, whereas in Zellballen pattern, fibrovascular stroma. This is an example of nest in malignant melanoma where you can identify this type of nesting pattern in the malignant melanoma. So, polygonal cells with fibrous septa and variable size nest. Here also you can identify these are the nest and having connective tissue in between the nest. So, the tumors showing nesting pattern are neuroendocrine tumors, melanoma, paraganglioma, adrenal cortical adenoma and nested urothelial carcinoma. So, important difference between Zellballen and nesting pattern is Zellballen having perfect rounded nest, rich vascular septa and sustentacular cells, while nesting pattern they are rounded island, less vascular and no sustentacular framework. So, all Zellballen patterns are nest, but not all nesting patterns are Zellballen. Now, let us see third pattern, trabecular pattern. So, trabecular pattern means tumor cells arranged in elongated cords or plates and separated by vascular sinusoids. So, here you can see these are the elongated uh, cords or plates which are separated by vascular sinusoids. So, this is trabecular pattern and these look like railway track pattern or brick wall pattern or lever plates will show trabecular pattern. So they are having long parallel chords. So here also you can identify trabecular pattern, and in high power view the cells are polygonal, abundant eosinophilic cytoplasm, rounded nuclei and separated by this vascular channel. Now how will you identify this vascular channel? These vascular channels are lined by endothelial cells. So these are endothelial cells. So they are spindle shaped cells lining these sinusoids. So this is a sinusoidal space, right? And this is the trabecular pattern, right? Now the tumors which show this pattern are mostly hepatocellular pattern, adrenal cortical adenoma, adrenal cortical carcinoma, neuroendocrine tumors, and if you see in tumor, mostly this hepatocellular carcinoma will show trabecular and sometimes pseudoglandular pattern. So here these are the areas showing trabecular pattern. So here you can see plates of cells are present and they are surrounded by sinusoids. So this is endothelial cells and these are the tumor cells. So this is trabecula, this is sinusoidal area and endothelial lining is there. So you can see along with this trabecular pattern there is some pseudoglandular pattern and gland layer architecture is there which shows dilated bile canaliculi. So here distinct cell border and cells are having round in shape nucleus, prominent nucleoli, eosinophilic and granular cytoplasm. Now fourth pattern is organoid pattern. It is a highly organized architecture resembling normal endocrine organs. So here you can see this organoid pattern recreate the normal architecture of organ from which they arise, right? So mainly they are showing a normal organ like pattern like glands, acini, ribbons, nest or trabecula in an organized fashion within one tumor. So whenever you see nest, trabeculae, acini, ribbons within one tumor, it is called organoid pattern. So in low power view, you can identify multiple repeating structure, uniform spacing in between this, and rich capillary network in between this nesting pattern. So it is called organized appearance. So most common example is pancreatic neuroendocrine tumor. Here you can identify these are the organoid arrangement in the form of nest, and here this is the ribbon, right? And if you see the tumor cells, it is having salt and pepper chromatin pattern and granular pink cytoplasm. So organoid pattern means all the patterns, repetitive patterns in an organized manner. Now why important? This organoid architecture mostly suggests neuroendocrine differentiation. So it is seen in neuroendocrine tumor, pituitary adenoma, parathyroid adenoma, pancreatic NET, and carcinoid tumors. So histological clue, when you see uniform nuclei, salt and pepper chromatin, minimal pleomorphism and delicate vasculature. So it is suggestive of organoid architecture, and mostly in the endocrine organ you will identify this type of pattern. Now fifth pattern, solid sheets pattern. So as the name suggests, solid means large confluent sheets without gland formation, without papillae or nest or trabeculae. So in low power view, you will see entire field filled with the cells, no architecture, minimal stroma and diffuse appearance. So here you can identify this portion shows only a large area showing arrangement of cells. This is called solid sheet pattern. And here in this area you can identify these are the having glandular architecture with A which is having lumen. So here this is solid sheet pattern and this is glandular pattern. So here also you can identify these are the tumor cells having no any organizing or any identifiable pattern, and cells are arranged in a sheet pattern, and in between you can identify these are the small lymphocytes. So these are the tumor cells and these are the lymphocytes. So diffuse proliferation, no lumina, no fibrovascular core, minimal collagen and may show necrosis. So common tumors, seminoma, diffuse large B cell lymphoma, poorly differentiated carcinoma, medullary carcinoma, embryonal carcinoma or anaplastic tumors, right? In which you will identify solid sheet pattern. So solid sheet should be distinguished from nest pattern, which is discrete island syncytial where indistinct cell border, diffuse infiltrative pattern in which cells invading normal tissue, and all these five patterns are seen in different tumors. So remember Zellballen means cell walls, vascular septa, sustentacular cells, and most common pheochromocytoma and paraganglioma. Nesting, islands in fibrostroma without the classical Zellballen framework. Trabecular pattern which show cords separated by sinusoids. Think liver or adrenal cortical tumor. Organoid, combination of nest, trabecular, ribbons, acini with uniform neuroendocrine cytology. And solid sheet, diffuse tumor, growth lacking recognizable architecture and often seen in high grade neoplasm or seminoma. So this is about all these five patterns of epithelial tumors, and you will identify this pattern now easily, and whenever you see this type of pattern you go for further IHC to differentiate the type of tumor. So in next topic we will discuss another five patterns like syncytial, alveolar, acina, tubular and cribriform pattern. So till then keep watching all the episodes, and I hope you will now easily diagnose Zellballen pattern and all these different pattern. So if you found this video helpful, please like, share and subscribe this channel for more such videos, and we will discuss all the patterns in different episodes one by one. So thank you for watching. Have a nice day.